Welcome to How Science Happens, a podcast by Wally Paxton, DJ So, and Doug Tree, professors at Brigham Young University. In the podcast, we bring you stories of cutting-edge science as told by world-class scientists and engineers from around the world who are on the front lines doing the work. We explore the highs and lows of discovery and what makes science such an exhilarating and frustrating process for those who do it. And because we're nerds, maybe we'll even learn a little science along the way.
Hey everybody, welcome to another episode of How Science Happens. I'm Wally Paxton and I'm here on Zoom with my friends Doug Tree.
Hey everybody.
And DJ So.
Hey everyone.
And we're also here with one of my friends, Professor Nate Wittenberg from the Department of Chemistry at Lehigh University in the beautiful Lehigh Valley of Pennsylvania. Hey Nate, it's good to have you here.
Good afternoon. Nice to join you.
All right, so just by way of introduction and to give you a little overview of Nate's story today, Nate earned his bachelor's degree in chemistry from the University of Minnesota, and then a PhD in chemistry from Penn State, where I met him. And then after that, went on to complete some postdocs at Edinburgh in the UK and University of Minnesota. He was then a research fellow at the Mayo Clinic in Minnesota for a little bit and published lots of papers, did some amazing science, and then returned to academia, sort of maybe not returning as we discussed previously, but back to academia as a professor in 2016 at Lehigh University in the Department of Chemistry. So we're so excited to have you, Nate.
All right, well, happy to be here.
We got some questions for you, as you know. First of all, one of the things that I like to find out about is like how you kind of got started in science. Like, was this something that you were doing when you were a kid, or is this an interest that you developed in high school or later?
Probably when I was a kid. I was always interested in science in school. So, that definitely helped develop my interests. I also, this kind of goes to where I grew up and what my background and things like that. But I grew up in a college town in Wisconsin, in Eau Claire, and there's a university there, a branch of the University of Wisconsin. And in the summer, they would have kind of, summer science camps sort of things. And I did that for a few summers when I was young. It was, mostly kind of outdoorsy, wildlife biology sort of things, going down to a little stream kind of by campus,
field trips.
Kind of on field trips, but it was walkable from the university campus, and we'd go out and collect different things and then bring them back to the classroom and talk with the professor and talk about what we found and why they were interesting and stuff like that. And so from there, it just kind of stayed with me that I was always interested in science, and I did okay in it in school. So, that helped. Definitely.
Yeah. So we talked just briefly before. You did an internship in, this is when you were in high school?
No, it was when I was in college.
Okay.
Yeah.
So let's get to college first. So you're in Wisconsin, and then you got to make some decisions about college and majors and that kind of thing. So what were you thinking about? What was on your mind?
I suppose unlike a lot of high school students now, I applied to one college. That was it. I sent my ACT score to one place. I knew I was going to go to University of Minnesota. I visited the campus a couple times while I was a high schooler, actually go to hockey games there. And I knew some people that went there, knew some alumni, and Wisconsin and Minnesota have a very nice agreement called tuition reciprocity, where even though I grew up in Wisconsin, I could attend a university in Minnesota for essentially in-state tuition.
Nice.
Vice versa for Minnesotans, they can come over to Wisconsin and do the same thing.
I thought that they don't like each other. That's what I thought.
Well, I think, the state governments get along to a degree where,
But the
so they'll let,
football teams might not like each other,
yeah,
DJ,
the
but
whole
that's
Packer
okay.
Vikings thing and the Gophers Badgers. Yes. So there's rivalry there, but I think Minnesotans and Wisconsinites are a lot more similar than they might readily admit.
Yeah. All
Yeah,
right.
so full disclosure here, Nate. I went to the U of M for my PhD, and looking at your timeline, it looks like we may have overlapped when you were postdoc in chemistry. I was in chemical engineering in
Okay.
my grad work, but anyway.
Nice.
yeah, great place. So you studied chemistry though.
I
You know,
did.
you talked about some ecology as a student, before
Yeah,
college.
just as a youngster. Yeah.
So
Yeah. So,
what changed your mind, or
well,
was this just part of the progression?
yeah, it kind of was part of the progression, I guess. I really enjoyed my chemistry classes in high school. Had a couple great teachers that made it easy to learn and made it interesting. So,
And that makes a huge difference. Yeah.
yeah. And again, it's one of those things that I did pretty good in the high school level chemistry. And so, when the time came I picked Minnesota for school. And then when I got there, you meet with an advisor and they say, well, what do you want to do here? Right? You can do anything. This is a giant university. What do you want to do? And I said, well, I want to be a dentist. And they said, well, you can't major in dentistry. And so they said you can be on a pre-dental track, but you need to, kind of pick a major, of course, of study at this point. I said, well, I did, pretty good in chemistry in high school, and I seemed to really like it. And they said, okay, we're going to start you as a chemistry major. And that's kind of where I started.
And then you stayed there.
I stayed there. Yeah, actually, I switched colleges though. At Minnesota, you could get a chemistry degree from the College of Liberal Arts, or at the time it was called the Institute of Technology, and now it's the College of Science and Engineering. And I was in the College of Liberal Arts, but I switched to the Institute of Technology really for no other reason than it got me out of the foreign language requirement.
All right. These are decisions. These are things you have to figure out.
Yeah, it got me out. There's nothing wrong with studying foreign languages, of course, and I think it can be very beneficial. But in terms of fitting things into my schedule, it would have been a little trickier. But I did have to take a couple more what were called technical electives at the time, and so I took a couple statistics courses.
Yeah. And that's so some places math qualifies as a foreign language requirement.
Yeah, I can
so,
see that, or programming,
math, statistics, programming, right?
Programming. Yep.
Yeah,
for sure.
Yeah. All right. So while you were there, now I think we got to the internships. So you had a chance to do some internships. So tell us a little bit about those.
Yeah. So in addition to doing my undergraduate research, which we can talk about too, during the summers, my last 3 summers, because it took me 5 years to get out of there, I worked at 3M. So, 3M is this massive conglomerate company that does just about everything and make products that are in products that you buy, and you have no idea that they're a 3M
and one of
product.
those M's stands for Minnesota. So
Minnesota Mining and Manufacturing, right? It started as a sandpaper company, right, or abrasives, and of course, to make an abrasive, you need an adhesive, right? And so, a lot of people know 3M from Post-it notes and things like that, but they make a lot of other things too. And so they have this technical aid program, which is what I was. And it's essentially a program for science students that are in their bachelor's studies, and they can work at 3M generally alongside a PhD level scientist, and sort of learn the ropes of industrial research. And it was a lot of fun. I learned a ton of stuff. Basically I was, the PhD, he was a wonderful mentor. He ended up writing letters for me for grad school, which was super nice. And he really helped me a lot in terms of growing as a scientist and just kind of keeping me curious, right? It wasn't like he gave me just kind of a recipe, cookbook recipe. He would some days, but then other days he would, leave a few things out or let me kind of make some modifications on my own.
Mm-hmm.
And then, if I did something that he didn't think was quite right, then he would kind of course correct me before I kind of went too far on a tangent.
Yeah.
So that was really nice. I did that for 3 summers. And, during my last year as an undergrad, I didn't have classes every day. So one day a week during the week, I would actually go in and work at 3M as well during the school year. So it was really nice. It paid okay. It wasn't like an unpaid internship, certainly.
Right. Yeah, that's nice.
But it was a really good experience.
All right. And so through this, you decided not to go to dental school,
Yeah.
and then and went to graduate school. So was this like undergrad? Did you apply to one place, or did you have
No,
others on your radar?
I applied to a few, right? But mostly places that had kind of I would say a reputation as being strong in analytical chemistry.
Yeah.
And I got in just like a lot of us probably. You get into some and you don't get into others, right?
Right. Yeah, that's right.
you gotta shoot your shots. And so, I took a few campus visits, which is a wonderful thing that a lot of graduate programs do. So if there are any prospective graduate students watching this, if you get into a place and they offer you a visit, absolutely take it.
Take advantage of it, right?
Even if you're not 100% sure that you'd even want to go there, right? It's a chance to visit a different part of the country, meet some new students from elsewhere, and who knows, you might click with people that are there. Or the faculty. And so I did a few of those. I really liked my visit at Penn State and liked the people I met there, the faculty and things like that. I had a really great student host who made my visit fun. And that's how I kind of picked Penn State.
So then you get to Penn State, and now you've got the next decision, which is how are you going to figure out which group you want to join?
Right.
So did you have a few people in mind, or did your host convince you to join their lab?
Well, I ended up joining their lab, the lab that they were in. So I talked with a number of people on my visit, before I decided on Penn State, and then after I decided too. I was kind of torn between, like, do I want to do biological analytical work, which is what my undergraduate research was at Minnesota, or do I want to go into more inorganic or organometallic or materials type things, which is what I did at 3M. So, I met with people that were doing research in a few of these different areas, and ended up settling on a what I thought was a really good group doing bioanalytical work. Yeah.
Okay. And this was Andy Ewing's group?
Yeah, Andy Ewing. It was my PhD advisor. He's since moved on to the University of Gothenburg in Sweden. But it was a really great group to join, and it was rather large at the time I joined it. There were maybe, I don't know, somewhere between around 10 to 15 graduate students. And a good handful of undergrads. So, and it had a real diversity of like projects going on. There were people doing a lot of mass spectrometry. There were people focused on different types of separations techniques, like capillary electrophoresis or chromatography. There were people doing electrochemistry. I ended up more on the electrochemical side, but my project ended up being different in some ways than anything that was going on in the group at the time.
Okay, so tell us a little bit about what you did there.
Yeah, so this project, it kind of got me started down the road of working with lipids and thinking about membranes. And so at the time, and still currently, one of the things that that group is working a lot with is studying neurotransmitter secretion. So this process of exocytosis. And a lot of the neurotransmitters that cells release can be detected electrochemically. Okay, so you put it's kind of crazy to think about. You have a single cell in a culture dish, and you can put a very small 5 micrometer diameter carbon fiber electrode on top of the cell, and you stimulate it, and it releases little packets of neurotransmitter. And these can be detected with the electrode. And so what my project was focused on was, I did use the electrochemical detection, but it was more along the lines of can we make something that releases molecules like a cell, like exocytosis, but is not a cell. All right. And in doing this, basically, if we just have the membrane part of the cell and not the protein machinery, can we sort of decouple the influence of the proteins and the lipid membrane? Like, can the lipid membrane itself, its sort of biomechanics, can they facilitate this process of neurotransmitter release?
Yeah.
And so we did that with something that are called giant vesicles, which are, it's a lipid bilayer membrane that's a kind of a large, well, relatively large sphere, you know, between 10 and 50 micrometers in diameter. And we'd poke and prod it with these small micropipettes, and it end up inflating small little lipid vesicles inside of the giant vesicle. And we'd fill them with something that was like a neurotransmitter, and then we'd have an electrode on the outside, and when they would fuse with the giant vesicle, we could detect the release of these neurotransmitter analogs. So it was a really fun project, because people in the group were working on electrochemical detection of exocytosis. I think my project was kind of the first, or I was the first person to tackle this idea of doing it, you know, in a non-living system.
Right. You successfully mimic the cellular process.
So
me and I did have some really fantastic help in that project. I can't say it was like me alone, because it certainly wasn't. And I think you talk to any scientist, they'll tell you it's never a individual endeavor, right? It's not
That's right.
You're not toiling in a lab by yourself. But yeah, I had a fantastic collaborator who came over from Sweden to kind of show me some of the methods and stuff like that, and it was wonderful.
So I mean, did it work? So basically, when you provide electricity, did it release something from inside?
Yeah, so we did. We used
okay.
We didn't use dopamine or norepinephrine. We used catechol. So you know, dopamine is a class of molecule called a catecholamine, right? But we used catechol, so it's just a benzene ring with two hydroxyl groups. Yep. So we would put that inside the small vesicles, and then watch them fuse.
Then those internal vesicles would fuse with the giant membrane, and then they would
dump out
the contents outside.
Okay.
Gotcha.
So interesting.
So
similar
beyond
to the way,
sorry, go ahead.
I was just going to say, this is similar to the way that your neurons work, where they have vesicles that will fuse and then spit out a bunch of neurotransmitters. Is that how it works, or is it
more or less?
Okay.
More or less. I mean, I think it can be somewhat more complicated than that, because in a cell, right, you have this protein machinery that really facilitates the fusion of these two membranes with one another, because they don't spontaneously fuse. So you need proteins that on the neurotransmitter containing vesicle, and proteins on the inner side of the cell membrane. They come together and make a little complex. It's called the snare complex, and they have different ion sensors, that, especially calcium, that trigger the change in conformation, and then this drives the fusion. It makes a little pore in between these two membranes. And of course, with the pore being open, neurotransmitter can leak through there. it doesn't have to fully open for it to get out. And
Okay.
so, yeah.
Well, this sounds like this could come with some challenges, as most PhD projects do. Was there a time when you just kind of felt like you were stuck and
Yeah.
needed to get
Yeah.
get
So,
over a wall?
yeah. So the first way we tried to do this was to try to, we had this big sphere and we tried to put the electrode inside the sphere
Uh-huh.
and then like pull out a little vesicle outside of the big sphere and then let it go. And then it would come
Uh-huh.
in and fuse.
Uh-huh.
It turned out that was really hard to do because the electrode was, rather large and not
Yeah.
sharp.
Mhm.
So what we ended up doing is leaving the electrode on the outside and sticking a very sharp glass pipette into the giant vesicle. So that went on the inside.
Mhm.
And then we could grab a little bit of the vesicle, the giant vesicle membrane, and sort of inflate these small vesicles from it.
Gotcha.
So that we tried to stick this electrode in for a long time. And we'd, tickle this giant vesicle with electric pulses and things like that to sort of temporarily destabilize the membrane. And we still couldn't get the electrode in. So
Mhm.
until we figured out like, hey, let's just stick this sharp pipette. We have it here anyway. Let's just stick that in.
Mhm.
That worked much better.
Well, this is I'm going to move on a little bit. So you got to a time in your PhD program where they say, now you are Dr. Wittenberg. Go
Yeah.
out and do amazing things. So so what happened there? What was the transition like from completing your dissertation to moving on to the next thing?
Yeah. So it's a tough time finishing up in some ways because you have to make this transition. Do you kind of what career path do you want to take? You don't have to make that decision immediately, but it should be on your mind at some point.
Yeah.
I think. And so I was pretty sure that I wanted to work in academics at some level. So I was thinking a postdoc would be right for me. And so, I kind of did the traditional thing of trying to email people that I'd had contact with, other faculty members at other institutions, emailing people that I had never had contact with, and just asking them if they have
the cold calls. Yeah.
Funding for a postdoc. And a lot of those go unanswered, those emails. It's normal.
Yeah.
But then I ended up going to Edinburgh. That was kind of one of these things where you see a job ad in the back of Nature, right? Or the Nature Jobs website.
Yeah.
And it's like, we have a postdoc position for someone to do electrochemistry, biologically oriented. There wasn't a whole lot of detail about the project. Looking for a chemist, right? Who knows electrochemistry and biological stuff. And so I sent my CV and had a couple phone interviews, on speakerphone. This is the days before Zoom, of course.
Yeah.
Like 2006. And I got the job and went over there for a little bit.
All right. So you're there in the UK for a little bit. This is about a year.
Yeah, a little bit less, but yeah.
Okay. And then came back to the States.
Yeah.
To University of Minnesota.
Yeah. So, it was a place that
coming home a little bit.
Coming home a little bit, a place I knew well.
Yeah.
I was looking to come back to the US and just reached out to some people that I knew there. Asked if they knew of anyone looking for a postdoc, and was able to luck out and join a fantastic group there in the chemistry department. That was Christy Haynes's lab. Another, fantastic mentor. And spent a couple years working in her group. It was a lot of fun.
Okay. All right. And then at some point there was this transition from the chemistry department to the electrical engineering department, which I think is kind of an interesting move for a chemist to make.
Yeah, yeah. So that was an interesting move, but it turned out pretty good. So in the group in the electrical and computer engineering ECE department,
Uh-huh.
they made a lot of optical biosensors,
Okay.
right? Or optical sensors in general,
Uh-huh.
right? And fantastic group of people in nanofabrication.
Uh-huh.
But to make a good sensor, you want it to be selective in one way or another, right? To detect the molecules that you want it
Right.
to detect and not other things. And so, one of the ways you can do that is by functionalizing the sensor in some way,
Mm-hmm.
right? If you have a sensor like the ones we worked on that detected changes in refractive index, right? Well, any biomolecule that sticks to the surface causes a refractive index change.
So you can
Anything.
detect biomass,
Yeah, you can detect
all
anything.
kinds.
Well,
Yeah.
you detect everything. It's kind of,
Yeah.
it's without the proper surface chemistry, these sensors are kind of, I mean, they're dumb in a way, right? They just,
Yeah.
if something sticks to it, you get a signal change, right?
Right.
And
Right.
so this group was looking for someone with lipids and membranes expertise because they
Uh-huh.
wanted to make a sensor that could detect lipid protein interactions,
Mm-hmm.
right? And this is kind of where the collaboration with Mayo Clinic comes in, because that the group in electrical engineering was collaborating with the group at Mayo Clinic. And the Mayo Clinic group had discovered a number of human antibodies that stick to membranes of nervous system tissues
Mm-hmm.
and can have interesting potential for treating various neurological disorders. And so what they wanted to do is make a sensor that could sort of characterize interaction kinetics and affinity for these antibodies to different lipids. And so that's where an electrical engineering group gets involved, as they have the sensors expertise. And then you
Yeah.
have the medical people who can supply all the antibodies and tell us the relevant biomolecules that we might want to put on the sensor.
Mm-hmm.
And then I came in by putting the molecules on the sensor, right?
Yeah. Okay.
So yeah, that's kind of how the transition worked.
Bringing those two worlds together.
Mm-hmm. Yeah.
Well, yeah. So that one of the things that I've noticed in talking to a lot of scientists is that the career paths never seem to be like a straight line. It's not like I did this and I did this and then I ended up exactly where I wanted to be.
Right.
So I'm
Right.
seeing that a little bit with yours too.
Yeah, and I was in Minnesota for a long time, right? I went there first as a postdoc in like 2008, 2007, something like that. And then I started here at Lehigh in 2016. So I was there for a while.
Mm-hmm.
It was a good time. The science was good. And at some point I just felt like I needed to give this a shot on my own.
Yeah.
Mm-hmm.
Yeah. Well, and good for you. You did it. And so now you're at you moved from Minnesota to Lehigh
Yep.
to start your own research lab.
Yep. Went, Minnesota to Pennsylvania to Scotland to Minnesota to Pennsylvania. So it's kind of like coming back.
So are you making an announcement in a few years to go to Scott? No.
No, I'm happy here. Not going anywhere.
Yeah.
So I came back here and started my lab in 2016. I was really fortunate in getting my kind of my lab off the ground, is that I was able to hire a postdoc right away. And
Yeah.
it was someone that I knew from my previous lab, right?
Mm-hmm.
And he had all the expertise that I knew I would need in the lab here, right?
Mm-hmm.
And he was, patient and meticulous, and I knew that's like, this is what you want when you're starting a lab, and
Yeah,
would
that's
be
right.
good for helping train new graduate students too.
Yeah.
And some of the kind of protocols and sort of standard operating procedures that we use in the lab were developed by him, and it's just kind of we've tried to keep it constant.
Yeah. All right. Well, I think that leads us to this paper that you sent to us that we want to talk about a little bit. So I mean, we've seen a lot of this background with electrochemistry and bioanalytical chemistry and lipids, and so then we have this paper, Nano Arrays of Individual Liposomes and Bacterial Outer Membrane Vesicles by Liftoff Nano Contact Printing. So the first thing you got to do for us is tell us what the words in that title mean.
Okay, I can do it. So nanoarrays, basically, if you think of an array as sort of a assemblage of things on a surface, we'll say. And the nano, of course, is a prefix meaning 10 to the minus 9. So this refers to the length scale of things, components of the array. So we're talking about objects that are less than 1 micrometer in size and less than 1 micrometer apart. Okay, so that's the nano array part of it.
Gotcha.
And so I have the paper here. I need to, individual, of course, everyone knows what that is.
Yeah.
Single objects, right? An array of single objects. And a liposome is a spherical structure composed of a lipid bilayer membrane. And, they can range in size from tens of nanometers in diameter up to maybe a couple hundred nanometers in diameter. The definition of liposome or small vesicle, I don't know where one ends and one begins, but they're small. They're composed of a lipid bilayer membrane. They have it's filled with water in the center, so you can trap different cargo in there if you want.
Yeah, sometimes I describe them to students as little tiny water balloons. So it gives them, there's a membrane. It's got some, it keeps it stuff inside, stuff outside.
It's a very small compartment. That's exactly right. And they're flexible, and they're made of lipids. So in some respects, they can be used or thought of as models for cellular membranes, albeit very simple ones.
Yeah.
And then bacterial outer membrane vesicles. So these are small vesicles that gram-negative bacteria, so bacteria with 2 membranes, bud off from the outer of the 2 membranes. All right. So they bud off. They're involved in bacterial communication with one another. So if you have an important cargo that you'd like to share with your neighbors, but you don't want it degraded by the environment, it would be helpful to put it in some sort of container. So that is one of the things that the bacteria do with them. And they also will use them to package toxins and things like that for infection. And the ones we work with are in the latter category. They have a toxin associated with them.
Okay.
And so those are the outer membrane vesicles. Then Liftoff Nano Contact Printing. So this is one word altogether, I would call it. Basically, if the listeners know what a rubber stamp is, right? Yeah. It's like have a stamp and you put some ink on it and you stamp it down on your envelope and it's your return address, right? Well, that works really well, even to pattern biological molecules on a surface, down to feature sizes that are maybe 500 nanometers or so. So very small. It can be shrunk. It can be miniaturized quite a bit. But once you start going smaller than that, there's some problems with the standard printing process, which is called micro contact printing. And our way of doing it, which full disclosure, we didn't invent, but we adapted it to our needs, is this lift-off nanocontact printing, and eventually, or essentially what it does is allows you to print biomolecules from one surface onto another without using any raised features in either substrate. So it's like a flat pattern getting transferred onto a flat surface.
Gotcha.
And this is important when you start shrinking things down, below 500 nanometers or so, just
Mhm.
because of, if you think of a rubber stamp, right, the rubber is flexible.
Mhm.
And if you have very fine features, that rubber can bend even under the weight of the stamp,
Mhm.
right? A 500 nanometer wide piece of a soft rubber is going to bend under,
It's gonna collapse. Yeah.
a couple grams that the stamp may weigh. So
Yeah.
we adapted this this method that was developed by a group in Canada to pattern biomolecules that could be used to capture liposomes and bacterial outer
Okay.
membrane vesicles. Yeah.
Gotcha. So you're
That's-
using a flat stamp, and you're just patterning what you want to transfer onto another surface on that stamp.
Yeah,
Gotcha.
it's kind of a multi-step process
Uh-huh.
where we do start with a template that has raised and lowered features,
Mhm.
but we eventually end up with a pattern of biomolecules on a flat piece, essentially a flat piece of rubber
Mhm.
called polydimethyl siloxane. And then we can take that pattern and transfer it to a flat piece of glass.
Mhm.
So it's a flat to flat transfer process.
Okay. So now you've got these nano arrays of
Yeah.
biomolecules on a surface. What do you do with
Yes.
them?
So the biomolecules are on the surface, right? And the biomolecules we pattern are in this work were all different types of proteins. Some of them were bacterial toxins that bind lipids that we can put in liposomes.
Mhm.
Okay, so that was the first thing we did, is we made these little patterns of toxin that was like a hexagonal array. So if you look at it, it sort of looks like a honeycomb.
Mhm.
And each one of the little spots in the array is 200 nanometers or so in diameter. So they're really small. And the liposomes are less than 200 nanometers in diameter. And our goal with this is to have, for each protein spot on the surface, we capture one particle, okay, one liposome on that
Mhm.
spot. And the reason for that is that allows us to look at individual objects.
Right.
So why individual objects? Well, if you take a million of something that are not all the same, right? But if you analyze them sort of as a group,
Mhm.
whatever feature you're trying to analyze, you'll get a value that is the average of the group.
Which doesn't account for the heterogeneity of your sample.
Exactly, right? Imagine if half of your particles have 2 copies of a protein and the other half have 0 copies.
Uh-huh.
Well, the average have 1, right? None of the particles in your system may have 1, right? This is a very simple, non-realistic example.
Yeah.
But if you were to analyze them as a group, or what we call an ensemble, it would really obscure the properties of the particles themselves. So you have to look
Yeah.
at them individually, and it helps if you can look at many individuals at the same time.
Yeah.
So that's why we want to put them on a surface, and we want to get them close together, but not too close together, right? Because when you start putting 2 objects closer and closer together, once you start going near distances that approach wavelengths of light that you're using, those objects, you can't tell them apart anymore.
Uh-huh.
2 objects that are, okay, you can, but you need a special microscope.
Right. Right.
With a regular fluorescence microscope, it can be challenging. So
Do you also run into challenges of the vesicles fusing to each other? Or does that not?
maybe,
is that not a
but I'm not
consideration
sure.
if you get them close enough to each other?
Yeah, so when you talked about making a stamp, So
Yeah.
I think if you want to capture one, so if you like to have individual vesicles, that means the distance between molecules of the stem should be far enough, shouldn't be?
Yes. So in our stamp, the spots are about 200 nanometers in diameter, and their spacing is 700 nanometers.
Oh, okay.
Yeah. So
Okay.
yeah, and that size of the spots and their spacing is defined by a template that we use in the beginning of this patterning process.
Mm.
So this allows you to get, you have enough of the molecule localized in one area where you can get one vesicle stuck, but then the next place that a vesicle can attach to is far enough away that you
That we
can
can-
resolve the
Yes, we can
Mm.
tell them apart.
differences between
And then
those.
in between the spots, we treat the glass.
Ah,
So
okay.
so things don't stick.
Ah,
Yeah.
okay. Mm.
Yep.
So these vesicles should only stick where you have patterned these
Yep.
molecules with
Yeah,
this
that's
array.
kind of the
Gotcha.
idea.
Gotcha.
And so really it's one of those projects that I kind of conceptualized it a long time ago, and wrote about when I was writing my application materials for faculty
Yeah.
jobs,
Yeah.
and it actually worked. So
We're always pleasantly surprised when things actually work.
yeah, everything works on paper.
Yeah.
Um,
You could
at
draw
the-
some PowerPoint cartoons, everything works, right?
yeah, it's working in theory versus working in practice.
Yeah.
So we were able to pack these liposomes rather closely in these arrays, and then from there we take and get some outer membrane vesicles from bacteria, and depending on what sort of molecules we pattern on a surface, we can capture different types of outer
Mhm.
membrane vesicles. In this particular work, we worked with a collaborator in chemical engineering here at Lehigh, Professor Angela Brown, and her lab works with a pathogenic oral bacteria that I just call AA because its name is very long. Yeah, if you want to know what it is, you can read the paper.
Okay.
But so they have a couple different strains of this bacteria, and one strain makes a toxin and puts it on the outer membrane vesicle that it buds off, and another strain doesn't. So with these arrays, we could, use an antibody, which is a protein that binds the toxin, right? And so we could capture vesicles that had toxin, or we could use an antibody that bound other proteins on the vesicles
Mhm.
that are there regardless of strain, right? So we could capture vesicles from both strains of bacteria. And so then we, did a couple experiments to show that what we're seeing is actually individual vesicles,
Yeah.
individual outer membrane vesicles, OMVs. And so, beyond this and sort of what we're working on now, I don't know if you're going to ask where is this going in the future, but I
might not have to. You're going there.
Yeah. So what we're doing now is, and we just put out a preprint on this. So plug. We didn't use the patterning part of this, but what we were able to do is take outer membrane vesicles from this AA bacteria and look at them on a surface and do two things simultaneously, which is measure their size, so the size of individual vesicles,
Mhm.
and whether or not they have toxin.
Mm.
So the outer membrane vesicles from this bacteria have a really unique size distribution. Most particles have kind of what we would call a unimodal size distribution, right? So there's just one peak. This bacteria makes these vesicles, and it has two peaks in their size distribution. So there's a kind of a subpopulation of small ones and a subpopulation of large ones.
Mhm.
And so it was the hypothesis of my collaborator, and they had good evidence to believe this is true, that this toxin was on the large vesicles and not the small ones.
Mhm.
And so what we were able to do
Ooh.
by kind of arraying them on a surface, albeit in a different way and a much sparser array, was, simultaneously measure size and whether or not they have toxin.
Mhm.
How we measure size is a bit of an involved process.
I was going to
because-
ask because you already mentioned that we're below those wavelengths
Yes,
of light, and so it's
yes.
going to be difficult to
So-
resolve.
Think of a sphere, all right?
Uh-huh.
And think about a sphere that the surface is dyed, all right? So
Yeah.
it has a fluorescent dye in it.
Yeah.
And now, if that dye goes into the membrane that makes the sphere, with sort of a constant concentration, right? Or it's uniformly distributed amongst the vesicles, sort of number of dye molecules per unit surface area is the way to think of it.
Okay.
Well, then the number of dye molecules per vesicle is proportional to the surface area of the particles.
Yeah, so you're going to get brighter areas for larger vesicles, right? Because you got
Exactly.
more fluorescence per spot.
Gotcha.
Yep. Yeah. So we look at it's the intensity, but it's actually the integrated fluorescence intensity for
Yeah.
every spot. So
Yeah.
we take a picture and then basically
Sum up all the area under the curve of that
exactly.
intensity. Yep.
It's one of those things that when I took Calc 2, I'm like, why do I need to learn about these integrals? Like, what are they good for? I mean, I learned really quick when I was taking organic. I'm like, oh, you integrate these peaks in NMR, and that's why you do it. That's why a chemist would want to know about it. But then there are other reasons too.
Yeah.
And so it's like the area under the curve, essentially taking an image and transforming it into a 3-dimensional surface,
Yeah.
right? And so for each little vesicle, we can determine its integrated intensity. And then if we measure the diameter or radius distribution with an alternative method, right? The mean radius with the alternative method should be the same as the mean radius of the particle stuck on the surface,
Yeah.
right? So then you can sort of basically make, with a little bit of algebra, kind of a scaling factor
Mm-hmm.
that says, okay, if we take the square root of the integrated intensity distribution, that is proportional to the radius distribution.
Mm-hmm.
Well, if you know the
Yeah.
mean radius from another method, you can sort of map the two distributions onto one another.
Yeah.
We didn't invent that method either. That was a group in Denmark that showed this maybe 10 years ago or so. But we found it works well for us. And then we can label these vesicles with fluorescent antibodies and determine which ones have the toxin and which ones don't.
Yeah.
And what we found is that the likelihood of a vesicle or an outer membrane vesicle being toxin positive is much greater if it's larger. It's basically like there are no toxin positive outer membrane vesicles smaller than 60 nanometers in diameter. And there are no toxin negative vesicles greater than 220 nanometers in diameter. But in
So
the
you still
middle,
have an overlap.
it's an overlap, which is, you can't determine this any other way
Yeah.
except for looking at them individually.
Right.
Because if you can separate them by size, like there are a number of methods where you can get like size fractions.
Yeah.
But let's say you have one in the middle, like they're 140 nanometers, and you'd say, oh, there's toxin in that fraction. But what we show, at 140 nanometers, only half of the vesicles actually have toxin.
Yeah.
So
So it's the right way of thinking about this, probably, that you have some physical process that this bacteria is doing that's giving out, some polydisperse, but two populations around some mean, small and large, and there's probably some distribution around that, and likely the large ones are the ones that are that have this, toxin attached to it, and the small ones don't. And that's probably two processes that are generating these that are different.
It
But
might
sometimes
be.
those sizes maybe overlap,
Exactly. Nature is can be kind of messy, right?
Sure.
And I don't know the kind of the mechanism behind this. Not a microbiologist.
Yeah.
So it could be, the toxin being on the membrane doesn't let the membrane bend as much. So when it buds
Yeah.
off, it has to be bigger, right?
Mm.
Or that could potentially be related to the biology of the cell and the way, there could also be all sorts of mechanisms from the cell standpoint why that might be.
Yeah. And, I'm not a microbiologist. My collaborator knows way more about these bacteria than I do. But we don't exactly know how the cell makes different ones, and like how the toxin is more likely to get sorted to the larger ones.
Well, that's a cool problem.
Yeah,
You guys cracked it.
Well, we'll see how it goes, in the peer review process, right? That's
Sure.
always a fun thing.
Yeah, well, send us a link to the preprint. If it's okay with you, we can include that in the show notes along with this paper that you sent to us. And
Yeah,
so
absolutely.
people who want more information and want to see the really nice pictures, you got some beautiful pictures in here showing these arrays.
Yeah, and I
Be able to
kind
look
of,
at those.
I think we have even better ones, that kind of were taken either after or, when you're putting together a paper from hundreds of images, sometimes it's hard. You just pick one that looks good enough for now, and then you go back and you're like, nuts, I think we have a better one.
These are better.
Yeah, but no, I think we got good ones in there.
All right.
My student who kind of led that, Jenny Colley, she was fantastic and took a lot of pictures.
Yeah. Well, that's great to have students that are on these projects and making it happen. All right, so I've got our last question for you. So imagine you could go back. You are looking at colleges now, and maybe you're at University of Minnesota again, but you're not going to choose chemistry.
Yeah.
What are you gonna choose? Are you gonna go for that foreign language credit?
I don't know. I mean, I was thinking about this kind of while we were talking and even before, and I think when people go to school, they should definitely do something that's really interesting to them.
Yeah.
And can be a rewarding career, right?
Yeah.
And potentially impactful either to yourself or your community or the world at large. Right? And so, I go fishing a lot, and I love it. But there's so much about like the natural world and wildlife and nature that I don't really know about, and I always want to kind of learn more. So, maybe I would do something along that line. So, getting back to where I started with science, right? With these outdoor
the field trips,
collecting critters out in the wild. So maybe something that would get me outside more.
Yeah. Yeah, we do spend a lot of time inside as scientists.
Yeah.
But that comes with some flexibility.
Yeah. There are chemists though that do things that require field work.
Yeah.
Right?
Yeah, that's right.
My roommate at Penn State for a while, he had done his master's before coming up in like natural products synthesis, and he would have to spend during his master's some times out on a research vessel, and they would go scuba diving and like cut sponges up from the bottom of the ocean and
then you do some separations.
I think the boat had a little lab on it. They could do HPLC and some prep work on the boat.
Oh, nice.
I don't know if I would go in that direction, but maybe something might get me outside a little more.
All right. Well, thanks for visiting with us for a little bit today. Our guest today has been Nate Wittenberg from Lehigh University, Department of Chemistry. Thanks for joining us. We appreciate your time, and this has been a lot of fun. You can find out more about Nate's research by going to our show notes. We'll put a link to the preprint and to this paper that we've been talking about here. You've been listening to How Science Happens with Wally Paxton, DJ So, and Doug Tree. And as always, remember to subscribe to our podcast, give us a rating. This helps other people find the show. And you can also find us on Twitter at Happens Science. And we will see you next time. Thanks again, Nate.
Thank you.
Thank you for listening to this episode of How Science Happens with your hosts Wally Paxton, DJ So, and Doug Tree. For more information about the podcast, the hosts, or our guests, please visit our website at bit.ly/howscience. For additional comments or questions, we can be reached by email at howscience@byu.edu.